What excites me most about this field is the possibility to create something really unique and valuable for patients' lives and safety. The field has emerged. We are now able to simulate kinetics and toxicity. We're now finally moving into disease models, and that's, I think, where the next 5 to 10 years will be our focus on, to develop something that actually can bring value to patients. Hi, I'm Chris. I'm leading the MPSlabs business unit within ESQlabs. We are a dedicated team focusing on the development of digital twins for organ-on-chips, MPS, organoids and spheroids. Our goal is to translate in vitro results to the patient side. MPSlabs, we try to solve two main things. One is we want to use our digital twins to understand the actual in vitro data, so that we really understand what the systems are telling us. And secondly, we want to then use those data and translate it to humans for PK, toxicity and efficacy predictions that then really allow you to improve your decision making and have drugs that actually help patients at the end. It's a three-step process. We first have our app Atlas. It's a software tool that allows you to do exactly that. You can configure your chip designs, you can analyze results in vitro, and from those analyses, for example, you get relevant parameters that describe your kinetics or your toxicity or efficacy, depending on the question of interest. And you then can plug that into a human in silico model or a human digital twin to then simulate the potential kinetics based off of your in vitro results. We see two main advantages. The first one is that you are actually using human relevant systems. We all know that animals, or mice in particular, are not mini-humans. And with our approach, we actually target exactly those three R's. So we can replace, refine and reduce animal experimentation because the workflow is animal-free at the end. Secondly, the computational approach and the platform allows you to run what-if scenarios. It's quite easy to run different scenarios if a barrier is broken or if the kinetics of a certain biomolecule is faster or lower than expected. And you can use that to simulate something before you actually run any bioexperimentation at all. Work with Dynamic 42 and Bayer on this pregnancy model was actually quite interesting. We had a lot of bioexperimentation on individual organs and on the integration of those three organs to predict the safety of prednisone in pregnant women. The idea was that we use all of the nice generated bioexperimentation and put this into our workflow, analyze it, and basically answer a very simple question: what is the exposure to an unborn baby? And we could simulate that this actually was quite successful in the sense that the drug would be safe if given regularly to the pregnant woman. And that project clearly showed that the integration of MPS-derived data and computational models really can inform clinical decision-making. What excites me most about this field is the possibility to create something really unique and valuable for patients' lives and safety. The field has emerged. We are now able to simulate kinetics and toxicity. We're now finally moving into disease models, and that's, I think, where the next 5 to 10 years will be our focus on, to develop something that actually can bring value to patients.